Hi guys, Miss Francis here to talk to you about our new unit, which is all about mitosis and meiosis, or how cells reproduce. Would you please do me a favor and make sure that by the end of this video, you understand these vocabulary words? My goal is to have vocabulary words before every video or before every lesson to ensure that by the end of the lesson, you can check yourself. If you don't understand these vocabulary words, then you need to go back and revisit what we talked about. So scientists study living and non-living matter, but how do they know whether something is living or non-living? Well, scientists distinguish living things from non-living matter by if they have the ability to reproduce through cell division. Cell division is important for multicellular as well as unicellular organisms, but the function of cell division varies in these types of organisms. So for example, cell division in unicellular organisms is used for reproduction to produce offspring, whereas cell division in multicellular organisms is used for growth and development to repair and renew any cells that die. Now, cell division requires the distribution of identical genetic material, so there needs to be continuity. In other words, how do I get this identical genetic material to reproduce? So let's say I have this fruit fly, right? This piece of paper represents a fruit fly, and it's got 8 chromosomes. If I split to do cell division, now each one of my offspring only has 4 chromosomes. But if I take that same fruit fly, if I take their cells, which contains the DNA, which contains the 8 chromosomes, and I replicate those 8 chromosomes before I separate, well, now my daughter cells do have 8 chromosomes and are replications or duplicates of the parent generation. Also, the cell making exact copies enables minimal to no mutations from one generation to the next. So the genetic information that's found in organisms is known as its genome. That genome varies from prokaryotes to eukaryotes, which we've discussed before. In prokaryotes, it's a single long circular DNA molecule called naked DNA, because the DNA isn't wrapped around histone proteins, where in eukaryotic organisms, the genome consists of several linear DNA molecules, where that DNA is wrapped around histone proteins. Therefore, it is not naked, and it is referred to as a double helix. But now in a human cell, we have 3 meters of this DNA. So how are we going to fit all of this DNA into a cell's teeny tiny little nucleus? And what happens is our DNA coils and packages itself into chromosomes. Each eukaryotic species has a characteristic number of chromosomes. So one species varies from another species in terms of the number of chromosomes it has. As far as humans go, our somatic cells or body cells have 46 chromosomes, where our gametes, or sperm or egg cells, only have 23 chromosomes, half the number of the chromosomes in the somatic cells, which makes sense so that when the egg and the sperm join in fertilization, now that cell has the full set, has 46 chromosomes. So now let's dive into eukaryotic chromosomes a little bit deeper. Like we said, eukaryotic chromosomes consist of a long linear DNA molecule, where that chromosome contains DNA, where that DNA is made up of hundreds to thousands of genes, where those genes are made up of different combinations of nucleotides. Each gene has a specific location or a specific locus on the chromosome. So how is eukaryotic DNA organized? Well, as I go from my base pairs to my genes, all the way up to my chromosomes, what's happening is supercoiling. In other words, I'm going to coil and condense and then coil and condense and then coil and condense so that my chromosome is really tightly wounded. So the nucleosome is DNA wrapped around 8 histone proteins. Chromatin is that DNA histone protein complex, and that's only present in a non-dividing cell, which is during interphase. Now, after DNA duplication occurs, chromatin condenses, and now it is visible under the microscope and called a chromosome. So you guys have this worksheet. I just want to annotate it a bit. We're discussing eukaryotic chromosomes, which involves linear DNA that are wrapped around proteins. Those proteins are called histine proteins. Well, why does DNA condense into a chromosome? Why does it supercoil? Because it's easier to move during cell division and so that it fits into the cell's nucleus. Well, how exactly does DNA coil itself and wind itself up? So let's start at the bottom. Let's just remind ourselves that a nucleotide is the building block of DNA. And what does a nucleotide consist of? It consists of a phosphate, a nitrogenous base, and a deoxyribose sugar. Now those bases can be A's, T's, C's, or G's when we're talking about DNA. So the order of those nucleotide bases determines the genes. Here are our polymers of nucleotides, which is ultimately going to form our DNA helix. So here's your DNA helix, right? That's your double helix, your twisted ladder, which is just the polymer of DNA nucleotides, where the nucleotide is the monomer of DNA. If we move a little bit further down, so this is where my DNA helix ends. And now I've got my nucleosome. Your nucleosome is the DNA wrapped around those 8 histone proteins. So here's approximately where your nucleosome and your histones end. Then my nucleosomes start to coil, and I form a 30 nanometer piece of fiber, essentially, where it's just coils of nucleosomes. So those coils of nucleosomes get coiled even further and basically form chromatin. So chromatin are coils of your 30 nanometer fibers. They are coils of your nucleosomes. And these are present during interphase when there is no cell division occurring. Finally, we are supercoiled into chromosomes. So chromosomes are supercoiled. Sometimes it's just called coiled chromatin. This is a replicated chromosome where it is one chromosome but 2 sister chromatids. So even though this is one chromosome, it is made up of 2 sister chromatids. Unlike the chromosome to the left, where this is one chromosome and 1 sister chromatid. So this is unreplicated chromosome, whereas this is a replicated chromosome. Replicated chromosome will be present in prophase and metaphase and contains 2 sister chromatids. Unreplicated chromosomes contain 1 sister chromatid and will be present during anaphase and telophase. So each duplicated chromosome consists of 2 sister chromatids. Now where those sister chromatids connect, that center is called the centromere. During cell division, those sister chromatids are going to be pulled apart and then repackaged into the two nuclei at opposite ends of the parent cell. In summary, the genome in prokaryotes is a single long circular DNA molecule that's naked. It's got no histone proteins associated with it. Whereas the genome in eukaryotic cells has several linear DNA molecules, where it's called linear because it's going to look linear under the microscope. This DNA is wrapped around histone proteins. So if you imagine the DNA like yarn, and those histones are the spools that this yarn is wrapped around. Some main ideas that you should know: the organization of eukaryotic DNA. Chromosomes are made up of hundreds or thousands of genes, where those genes contain DNA that are made up of various base pairs. To elaborate on eukaryotic chromosomes, they are located in the nucleus. They are made up of chromatin that is coiled or supercoiled, and it contains your DNA and your histone proteins. Lastly, to help you fill out your first 5 vocabulary words, you can add this information to your chart. I just want to point out that chromatid and chromosome share characteristics. For example, a chromosome is also coiled chromatin and is also visible using a microscope.